Scientists say the universe started from an unbelievably tiny, insanely hot singularity. Then, boom! The Big Bang happened. Space-time and matter all burst out in one cosmic fireworks show. And just like that, the universe was born. Theologians, on the other hand, say the universe came from the will of God, out of nothingness. God said, "Let there be light," and there was light, followed by stars, earth, life, and eventually, us, humans. One sounds like it came straight out of a science lab, the other, more like a prayer echoing through a church. But if you're the kind of person who likes to think a little deeper, you probably asked yourself this: who put that singularity there in the first place? And if God created everything, then who created God? Keep chasing those questions, and you'll run headfirst into the same philosophical brick wall. What exactly was the starting point? In science, questions like this have a name. Unsolvable problems, not the kind where we just don't have an answer yet, but the kind where, logically speaking, there is no answer. Like, why does existence even exist? Almost everyone, at some point, has wondered. What's the meaning of life? It's a deep, complicated philosophical riddle, and there's no one-size-fits-all answer. Maybe it's about chasing happiness, creating value, finding purpose, or making a difference in the world. But here's the kicker. Have you ever stopped to ask, why do humans exist at all? How did we even get here in the first place? Darwin had a pretty classic take on it. Humans evolved from single-celled organisms to fish, to amphibians, to mammals, to apes. Then one day we're walking upright, making tools, and chatting away like it's no big deal. It's a wild ride of mutations and adaptations. That explanation pretty much ruled the Western biology world from the 19th century onward, until we cracked the genetic code. Even some Nobel Prize level scientists began raising their eyebrows, wondering, wait, how exactly did we get here? Which brings us to the story we're gonna dive into today. Let's rewind to Switzerland, 1869. A young medical intern named Friedrich Mischer, under the guidance of his mentor, was studying the chemical makeup of white blood cells. His daily grind, collecting pus from patients' wounds, then extracting the white blood cells from it. Yeah, I know, most people would know about just hearing about it. One day, during a random experiment, he dissolved the cell nuclei in an alkaline salt solution and ended up with a weird substance that wouldn't dissolve in acid. It wasn't a protein. It wasn't any known carbohydrate. It was a completely new kind of acidic material. Mischer had no idea he'd just stumbled onto DNA. All he knew was that it contained phosphorus. It didn't denature like proteins do, and it showed up in every single cell nucleus. He called it nuclein because, well, it came from the nucleus. Sadly, his work didn't make headlines. Nobody at the time thought the acid inside a cell's nucleus had anything to do with heredity. For the next 80 years, DNA was just some kind of acid to most scientists. Nobody knew its shape, and no one realized it was the ultimate storage device for life's blueprint. Back then, scientists guessed it might be a chain, maybe a twisted coil, or even just a messy ball of string. No one could say for sure, until one photograph quietly waited for its moment to change the world. And only then did humanity realize that this young guy, hunched over lab samples of wound gunk, had accidentally discovered the code of life. 1952, King's College, London. A brilliant scientist named Rosalind Franklin was deep into a tedious but laser-precise experiment. Her mission, using X-ray diffraction to photograph the crystal structure of DNA. One of those photos, labeled Photo 51, would turn out to be one of the most important images in the history of science. This blurry, mysterious image looked like a ripple stretching across time itself, spreading quietly through the darkness of the universe. From it, Franklin deduced that DNA might be a spiral with two intertwined strands, and that its phosphate backbone probably sat on the outside. But Franklin wasn't the type to jump to conclusions. Her scientific standards were high. She wanted more data before making a final call. Meanwhile, over at Cambridge University's Cavendish Laboratory, two young guns, James Watson and Francis Crick, were taking a completely different route. Experiments, not really their thing. They preferred to build models, literally piecing together atoms like they were playing with Lego, hoping to snap together the true face of life itself. They weren't getting anywhere spectacular until early 1953, when they happened to see Photo 51, courtesy of Franklin's colleague, Maurice Wilkins. Watson took one look and practically blurted out, "This is a helix." Crick's eyes lit up. "We just found the missing piece." From there, it was full speed ahead. They built a model that would rock the scientific world, the double helix, each step made of two base pairs, the helix winding elegantly around an imaginary axis, beautiful, precise, and perfectly symmetrical. In April 1953, Nature published 3 back-to-back papers. The first, by Watson and Crick, unveiled the double helix model. The second, by Wilkins' team, provided supporting X-ray data. The third paper featured Franklin's experimental results, including the Photo 51. It wasn't until the papers went public that Franklin realized her work had been leaked. Turns out that legendary photo had been used without her permission. Basically, it was the science world's version of a spy drama, all played out in lab coats. Morris and Franklin's time working together at King's College was far from friendly. Behind the scenes, he quietly handed Photo 51 over to Watson, who together with Crick quickly built their model and beat everyone to publishing in Nature. In that paper, Franklin was never clearly credited as one of the key contributors to the model's discovery. By the time she passed away in 1958, she still had no idea how her data had been borrowed. In 1962, Watson, Crick, and Maurice won the Nobel Prize for discovering DNA's structure without so much as a mention of the woman who took Photo 51. It took years before people fully realized the photo's true value and finally admitted that without Franklin's image, the secret of the double helix might have stayed hidden for many more years. Many scientists and historians have since spoken out on her behalf, calling her the forgotten fourth Nobel laureate and posthumously honoring her as the uncrowned queen of the double helix. But what's the use now? She's gone without her knowledge. That single photograph had already become the stepping stone for others to enter the Scientific Hall of Fame. As some say, the march of science sometimes carries the flaws of human nature, and all we can do is make sure the names once left out are finally written back into history. Ever since the DNA double helix structure was proposed, DNA research has become one of the main storylines in the modern saga of biology and medicine. In 1958, two scientists, Matthew Meselson and Franklin Stahl, ran a now classic DNA replication experiment. Using nitrogen isotope labeling, they proved for the first time that DNA replicates in a semi-conservative way. One old strand pairs up with one newly made strand. Fast forward to 1977. Frederick Sanger invents Sanger-Shijunning, letting humanity read its own genetic code for the very first time. Then in 1983, along comes Kary Mullis with his brainchild, the polymerase chain reaction, better known as PCR. This technique could take a tiny microscopic amount of DNA and amplify it billions of times over. PCR kicked off nothing less than a revolution in DNA copying. And here is where it gets wild. The idea for PCR didn't come from a quiet lab bench. It came to Mullis in the middle of the night, barreling down a California highway, listening to the radio. That's right. One headlight on the road, one head in the clouds. Mullis wasn't your typical lab coat type. He was a nonconformist hippie chemist who dabbled in psychedelic drugs. I've always wondered what song was playing on that radio at the time. Maybe it was Every Breath You Take by the Police, or more likely it was Hotel California by the Eagles. The timing checks out, the vibe matches, and those trippy atmospheric lyrics fit that night drive moment almost too perfectly. Moral of the story: everyone needs a hobby. Even one that sounds a little questionable might someday fuel a breakthrough. You never know when your bad habit will pull off a good deed for science. From 1990 to 2003, an international collaboration known as the Human Genome Project set out to decode all 3 billion base pairs in the human genetic sequence. Then, in 2012, came CRISPR, a gene editing tool that works like molecular scissors, letting scientists snip, swap, or insert gene fragments at pinpoint locations. By 2018, the world saw its first genetically edited babies. He, Jian, an associate professor at Southern University of Science and Technology in China, and his team edited the embryos of 2 baby girls so they would be born with immunity to HIV. The result? A global ethical firestorm. Humanity now had the power to rewrite genes, but also the responsibility to ask, should we? He, Jian, was later punished by law. In just a few decades, we've gone from decoding the mysteries of nature to editing our own genetic script as easily as editing a document. What we hold in our hands is no longer just a lifeless string of biological code. It's a holy manuscript, the book of life, telling the story of you. And the closer we look, the more it gives us goosebumps. This book is DNA, a long chain built from 4 chemical letters, A, T, C, and G, arranged in a sequence that can't be explained away as pure randomness. Together, they form a 3 billion character text. And that text is you, whether you are male or female, tall or short, brilliant or adorably. It's all written in those lines. Even more mind-blowing. This chain is coiled so tightly it fits inside a cell nucleus just 6 micrometers across. But if you stretched out one DNA molecule end to end, it would be 2 meters long. And every single cell in your body, tens of trillions of them, contains one of these 2-meter strands. Line up all the DNA in your body, and it could wrap around the Earth dozens of times. Kinda reminds you of those old-school punch cards from the early computer days, doesn't it? Back then, information was stored as a series of tiny holes, simple yet magical. DNA is basically the oldest punch card in existence, except it doesn't run on binary. It runs on quaternary code. Its 4-letter alphabet, A, T, C, and G, packs way more data per unit than the most advanced human-made hard drives. To put that into perspective, modern tech can store about 215 petabytes of data in just 1 gram of DNA. That's roughly 215 million gigabytes. What does that even mean? Well, the total global internet traffic in a single day is estimated at around 200 petabytes. In theory, 1 gram of DNA could store more than that. And here is the kicker. DNA doesn't just have insane storage density, it's also ridiculously long-lasting. Your average hard drive may be 5 to 10 years before it starts losing data. A DVD a few decades if you are lucky. But DNA keep it dry, cool, and out of the sun, and it can stay stable for thousands of years, sometimes even over 10,000. In 2013, a group of European scientists encoded data into a DNA sample, stored it under standard lab refrigeration, and 2 years later read it back perfectly. They calculated that at -18 Celsius, about 0 Fahrenheit, DNA could last over 100,000 years without losing a single bit of information. So if humanity ever wants to leave a message for some far-off future civilization, the safest USB drive might not be silicon. It might be a piece of synthetic DNA. Every person's DNA is split into 23 pairs of chromosomes, about 6 billion base pairs total. That's your complete source code. Scientists can now use it to detect family relationships, health risks, even certain tendencies in intelligence and personality. But DNA isn't just a passive storage device. It can self-replicate and self-repair. That's one of its real miracles. If part of the chain breaks or is copied wrong, DNA will call on the surrounding sequence to patch the error, something even modern computers struggle to do, and it explains why even tiny differences in chromosomes can have massive effects. For example, human DNA and chimpanzee DNA are about 98.7% identical, Yet one small difference in chromosome structure gave rise to two wildly different civilizations. Or take Down syndrome. Patients have an extra copy of chromosome 21, turning the normal pair into a trisomy. That's why it's also known as trisomy 21. Beyond simply studying it, modern science is itching to start editing this book of life. The He Jiankui incident was just the opening chapter, but here is the real kicker. Should humanity rewrite its own genetic script? And if so, who gets to hold the pen and meet the moral and ethical standards to use it? And it goes even further. Scientists can now literally store digital information inside DNA molecules using a method called solid-phase phosphoramidite synthesis. They can build artificial DNA strands and encode binary zeros and ones into sequences of A, T, C, G. We're talking wild stuff here. They've already stored the complete works of Shakespeare, and even the first few chapters of Harry Potter, inside DNA, and then read it back successfully. In other words, DNA isn't just God's language, it's also the most stable storage medium in the known universe. If future civilizations dig us up, they might not find pyramids. They might find a strand of DNA holding our entire story. Scientists have even tried extracting leftover DNA from dinosaur fossils. Most of it has, of course, broken down over millions of years, but in a few rare cases, in perfectly preserved amber, bones or teeth, they found trace genetic fragments. In Montana, for example, researchers discovered Tyrannosaurus rex bones with blood vessel-like structures inside, and within them tiny protein and nucleic acid remnants. We're not ready for Jurassic Park just yet, but these finds prove DNA is ridiculously stable as an information carrier. It's no wonder some people bluntly say, humans are basically software written in biological code. The catch: nobody knows who wrote it. And the code itself, it's suspiciously too good. Self-repair capabilities, double helix architecture, super compressed data storage, even baffling redundancy systems. Some sequences look like useless junk until you delete them, and then the organism dies. That's more mysterious than the most complicated computer code ever written. One British computer scientist, after studying DNA, said, "This doesn't look evolved, it looks designed." And that's not just a throwaway line. Even Francis Crick, yes, the Nobel Prize winning co-discoverer of DNA, began having doubts late in life. He and Watson were the ones who uncovered DNA's famous double helix structure. But as his research went deeper, he became more and more convinced that life's mind-boggling complexity couldn't be explained by random processes on Earth alone. So he dropped a bombshell of an idea, something he called directed panspermia. His theory life may have been deliberately seeded on Earth by some advanced intelligence from a faraway corner of the universe, and echoing what that British computer scientist once said, he admitted, "DNA just looks way too much like it was designed." In 1990, the United States kicked off with the UK, Japan, France, Germany, China, and others on board. A project so ambitious it was basically the moon landing of biology, the Human Genome Project. Its mission: cracked the entire genetic code hidden in human DNA. All 3 billion base pairs, like an epic, never-ending novel with no table of contents. And scientists had to read it letter by letter. Back then, genome sequencing was painfully slow and insanely expensive. At first, decoding just one person's full genome took years, and a jaw-dropping $3 billion. Some people even joked it was the world's most expensive jigsaw puzzle. But it was this very puzzle that finally gave us our first complete look at humanity's source code. In 2000, US President Bill Clinton, UK Prime Minister Tony Blair, project leader Dr. Francis Collins, and private entrepreneur Craig Venter held a joint press conference at the White House. They announced that the first rough draft of the human genome had been completed. In that moment, Clinton said, "Today, we are learning the language in which God created life." It was both a victory for science and an echo of faith. And here is the twist. The man leading the charge, Dr. Collins, was not just a scientist. He was also a devout Christian. Later, he would say that decoding the genome didn't pull him away from God. It made him more in awe of him. Collins led over 3,000 scientists working for 13 years to crack the instruction manual for humanity. Yet at the very peak of his career, he told interviewers, "Faced with the mystery of DNA, I can't help but believe in the existence of an intelligent designer." What makes this even more surprising? Before the age of 27, Collins was a staunch atheist. What changed him? He recalls that when he first saw the DNA sequence, its flawless logic, its elegant architecture, it filled him with a kind of reverence that felt almost religious. He began calling DNA the language of God, saying, "If the universe is a book, then DNA is the chapter God wrote for humanity." In his best-selling book, The Language of God, Collins proposed a framework to unite science and faith. He called it biologos. He believed God used the laws of nature, including evolution itself, to bring life into being. Not everyone was thrilled with that idea. Critics shot back, "Complexity doesn't require God. It just requires enough time." Their favorite example: if you give a monkey a typewriter and infinite time, eventually it will bang out the complete works of Hamlet. The problem: we don't have infinite time. The window for life to emerge was absurdly short. If DNA really evolved through pure randomness, then, given the age of our universe and the lifespan of Earth, there just isn't enough time on the clock. Harvard mathematician David Berlinski ran the numbers. Even if you take the Earth's early primordial soup, that chaotic mix of water, gases, lightning, and electrolytes, and shake it at the speed of one new combination per second, it would still take longer than the age of the universe to randomly cook up a usable DNA strand. Similar doubts haunted another Nobel Prize-winning scientist, cell biologist Christian Decoding. He believed life's emergence could be neither too fast nor too slow. Its timing was just right, almost as if it had been fine-tuned. On his deathbed, he wrote, "The universe seems to exist for the purpose of bringing forth consciousness. And remember, inside a real organism, we're not talking about a few pages of code. We're talking billions of lines of instructions with high fault tolerance, self-replication, and built-in redundancy. It's like taking apart a Boeing 747, scattering all the parts, and then, after a hurricane blows through, finding the plane reassembled and ready for takeoff." Sounds pretty wild, doesn't it? You might be wondering, wait, could our genes have been designed by aliens? Well, you are not alone, in fact. There is a whole crowd of people who've asked the exact same thing. There is even a theory for it. It's called panspermia. And actually, Francis Crick's directed panspermia is just a more specific version of that same idea. The core of the theory life didn't just pop up on Earth by accident. It was planted here by some kind of intelligent civilization. Sure, that sounds like something straight out of a sci-fi novel. But it also hints at something deeper. If even top-tier scientists believe the origin of life on Earth might not have been a random accident, then why stop at imagining aliens? Why not take it a step further? What if the designer wasn't an alien race at all, but a consciousness even more advanced, even more fundamental? We can't prove the existence of God, but we can flip the question around. If the complexity, purpose, and sheer information density of DNA exceeds what natural systems could ever realistically produce, could there be a mind behind it all? A mind older than the universe itself? There is another bold idea that's been gaining traction in some fringe corners of science. The notion that consciousness doesn't come from the brain at all, but from some kind of underlying field woven into the fabric of the universe itself. And in that model, DNA might just be the interface, a biological port that receives, stores, and expresses information from this consciousness field. You and I, we're not just living beings, we're information nodes. Being alive is simply running a program, which means we're not the ones writing the code. We are the code. And that consciousness, whether you call it God, the creator, Buddha, the source, may have written it all long before we ever showed up. But here is the thing, that consciousness doesn't have to be a bearded old man in the clouds. And it doesn't have to be some robed deity sitting on a throne. It could be a pure source of information, a source of purpose, a source of value. Science and theology don't always have to be mortal enemies. Science tries to answer how things happen. Theology wrestles with why they happen. Sure, DNA's structure can be explained with chemistry, but why does life even have the will to survive? That's a question from an entirely different dimension. Maybe, just maybe, one day we'll discover that the most advanced science and the most ancient faiths meet at the very same horizon. In the spiral of our genes, there may be an unseen hand, and behind humanity's birth, there may be an unwritten script. So, what do you believe? That we're nothing but nature's accident? Or that we're living verses written by a mind beyond our own?